We are going to be discussing compound leaf formation. And we're going to start with the development of compound leaves. A leaf that displays the division of a leaf blade into leaflets are considered compound leaves. Compound leaves consist of a petiole and several leaflets, and each leaflet can have a short petiole. There are different classifications of compound leaflet arrangement: pinnately compound leaves or palmately compound leaves. Pinnate leaves occurs when subdivision of a leaf into leaflets from the rachis. Palmate leaves occur when all the leaves emerge from a common point at the distal end of the petiole. Most common compound leaves exhibit bilateral symmetry, where one side is more or less a mirror image of the other side. There are three routes of leaflet production. The first one is acropetal succession. Leaf growth occurs from the base to the apex. Differentiation begins near the base and progresses towards the tip. So you'll find younger leaflets at the tip. The second is basipetal, which is the opposite of acropetal, and leaves are developing or opening in succession from the apex to the base. Younger leaflets are found at the base, while more mature leaves are at the tip. The third one is divergent, when leaflet development occurs at both ends without specific succession of production. So younger leaflets will be found near the tip and the base. The image shows the hypothetical stages in evolution of simple leaves from shoot systems and their subsequent elaboration into compound leaves. Compound leaf development arose independently many times during plant evolution. Therefore, there isn't one molecular mechanism that is representative of compound leaf formation for all plants. For example, in the image E, it is unclear whether it arose from an ancestor with lobe leaves or if it's due to an expression of specific genes. Compound leaf development varies dramatically among families and even within genera.
Next, we're going to discuss the biological significance of compound leaf development. As we've been discussing in class all quarter, nothing occurs in plant biology without a good reason. You might recall the example of Neobeckia aquatica from class earlier this quarter. Different environmental conditions can cause the exact same plant to have different degrees of leaf complexity. The leaves in highlight have simple leaves, whereas the submerged leaves have highly compounded leaves. Because these different degrees of complexity arise in different environmental conditions, we know that there are different advantages which are better suited for certain environments that come from having more highly compounded leaves. In this situation, the exact same species has different levels of complexity due to living in different environments. But as plant speciation has occurred, there have been multiple instances of compound leaves arising in different species. So now let's ask the question, why does having highly compounded leaves give plants an advantage in certain conditions? There were two hypotheses when it came to what individual leaflets were capable of doing. The first hypothesis was that an entire compound leaf was equivalent to a simple leaf in terms of function. If this hypothesis were true, then having a compounded leaf with many leaflets would likely not give the plant any advantage and would be a waste of energy to differentiate the leaves without providing any benefit. The second hypothesis was that each individual leaflet is functionally equivalent to a simple leaf. The second hypothesis was found to be true, and this can have advantages for photosynthesis. As we know, in most plants, leaves are the major light-gathering organ. And of course, we know that gathering sunlight is necessary for plants to carry out photosynthesis, which is the plant's process of synthesizing glucose from carbon dioxide, water, and sunlight. So as said before, since each leaflet can do the same function as a simple leaf, the more of those units a plant has, the more it can absorb sunlight and the more it can photosynthesize. This makes sense if you think about the Neobeckia example again. In the highly submerged plants, it's harder for plants to gather sunlight because they are underwater. So those plants benefit from being highly complex so they can gather more sunlight and photosynthesize more efficiently. Another benefit of having highly compounded leaves is that it can help a plant protect itself from herbivory, which is just the process of animals eating plants. Various studies don't necessarily agree that increased leaf complexity arose directly to defend against herbivory, but it may just be an unintended benefit. Whether this was an intended consequence of increased complexity or not, increased complexity certainly does help protect against herbivory, because if you think about it, it's more difficult for an animal to reach and eat small leaflets compared to taking a bite out of a large leaf.
Now we will describe the genetic and molecular mechanisms of compound leaf development. To begin, the class 1 knotted 1-like, or KNOX1 transcription factors are needed for establishing and maintaining the meristematic activity of the shoot apical meristem. KNOX1 genes act by positively regulating the cytokinin pathway, which is mainly involved in promoting meristematic fate, and by negatively regulating the gibberellic acid pathway, which promotes cell differentiation. The initiation of leaf and leaflet primordia and formation of the leaf margin require localized auxin concentration and pin-1 polarity. The KNOX1 genes are expressed within the shoot apical meristem, but they are excluded from the leaf primordia. For both simple and compound leafed species, leaf initiation requires the downregulation of KNOX1 genes at the incipient leaf primordia. Once initiated, leaf primordia will expand laterally. Within the leaf primordia, the asymmetric leaf 1, or ARP genes, repress the expression of KNOX1. This leads to high levels of gibberellins in the leaf primordia, which promotes growth and differentiation. Unlike simple leaves, however, in compound leaves, the KNOX1 genes become reactivated in the developing leaf primordia. Next, secondary structures begin to develop from specific meristem regions at the leaf margin. These areas are known as marginal blastozones. Within the marginal blastozones, the lanceolate, or the LA transcription factors, will function in restricting the activity of the leaf marginal blastozone. A specific microRNA, called microRNA 319, downregulates the LA transcription factors and enables meristematic activity of the marginal blastozone. The single leaflet 1, or SGL1 gene, which encodes for the flow or leafy type transcription factors, functions in the initiation of lateral leaflet development and leaf proximodistal axis development. A class M KNOX protein, called FCL1, is also required for the proximodistal axis development. SGL1 and FCL1 act in parallel pathways, and they are necessary for the development of leaf petioles. Another transcription factor, called PALM1, directly inhibits SGL1 activity to regulate its expression domain. In a diverse range of eudicot species, a NAM or CUC transcription factor functions in the development of lateral organ boundaries. The boundary gene described are necessary for the proper separation of leaflets. Finally, following the lateral blade growth patterning, two main pathways function to promote the separation of leaflets by specifically repressing the growth between them. These pathways include the low auxin response, as well as the expression of the reduced complexity or RCO transcription factors. To conclude, the mechanisms discussed represent pathways that have been found to be important for different plant species, and variations will be found depending upon the organism. The factors highlighted in red here have been shown to be conserved. Compound leaf development is a complex and a highly regulated process that provides fascinating insight into how complex organs arise from simple structures.